Hi, I am Ana Marie Alegria, and in this video, we will explore the intricate workings of the male reproductive system, where every organ plays a crucial role in the creation of life. So here is an overview of the location, structure, and function of the key organs of the male genital system. So, the male genital system, or also known as the male reproductive system, consists of several organs that works together to produce and deliver the sperm for reproduction. Now let's discuss the function, structure, and location of these organs. Let's start with testes. So here is the closer look of testes. So the testes are two small organs that are found inside the pouch of skin that holds and helps to protect the testes, called scrotum. So, what is scrotum? Scrotum is yung balat ng egg ng boys. Basta alam mo na, ayun yung balat ng ano. So, as you can see, the testes are oval shaped. Kung alam niyo yung itsura ng olive, parang ganun, tas pinalaki lang. Kung alam niyo olive, ganun yung itsura niya. So, ano nga ba yung testes? Ayun nga, kasabi ko lang, testes is egg ng boys. Ayun natin yung kapag nasangge or na parang nasaktan. Sinasabi nilang mababaw sila, ganun. O, ayun ata yun. So, ito na nga. Ano ba yung ginagawa ng testes? The testes are responsible for sperm production through spermatogenesis in seminiferous tubules and for the secretion of testosterone by the Leydig cells. So, ano ba yung ng spermatogenesis? So, spermatogenesis ensures the continuous production of sperm of fertilization, while the testosterone plays a crucial role on regulating male sexual characteristic and reproductive function. So, connected to the testes are the epididymis. The epididymis is a coiled tube located behind each testicle. Serve as a storage and maturation site for sperm before they are ejaculated. The epididymis is where sperm produced in the testes to gain motility and the ability to fertilize an egg. It also absorbs excess fluids and provides nourishment to the sperm. Additionally, the epididymis are responsible for transporting sperm from the testes to the vas deferens during ejaculation. Next is the vas deferens. The vas deferens, also known as ductus deferens or sperm duct, is a long muscular tube that connects the epididymis to the ejaculatory duct. Its primary function is to transfer mature sperm from the epididymis to the urethra during ejaculation. As the sperm travel through the vas deferens, they mix with seminal fluid to form semen, which is essential for fertilization. So next is the seminal vesicles and the prostate glands. Seminal vesicles are located near the base of the bladder and secrete a fluid rich in sugars and proteins that nourish and support sperm, while the prostate gland surrounds the urethra and produces a milky fluid that helps activate and protect sperm. So the seminal vesicles and the prostate glands works together to produce the fluids components of semen, supporting and nourishing sperm for fertilization. So now we're in urethra. Urethra is a tube-like structure that connects the bladder to the external environment. It is a tube that runs through the penis and carries both urine and semen. It serves as a passage to the body. And now we shift our focus to a key player of anatomy, the penis. The penis is the external male reproductive organ that is involved in sexual intercourse and urination. It contains erectile tissue that becomes engorged with blood during sexual arousal, leading to an erection. Overall, the penis is important for sexual function, reproduction, urinary health, and overall well-being. Its role goes beyond physical function and extends to aspects aspects of identity and overall health for men. Together, these components of the male genital system work harmoniously to ensure fertility and the continuation of the human species.
So, the Dux deferens, also known as the vas deferens, it is a crucial component of the male genital system. It is a muscular tube, approximately 30 to 45 centimeters long, that transports sperm from the epididymis to the urethra. Originating at all tail of epididymis, the Dux deferens ascends within the spermatic cord, which also contains blood vessels and nerves. It passes through the inguinal canal into the pelvic cavity, where it loops over the ureter and descends behind the bladder. During ejaculation, smooth muscle constructions in walls of the Dux deferens propel the sperm forward. The Dux deferens then joins with the duct with the seminal vesicle to form the ejaculatory duct, which passes through the prostate gland. Here, seminal fluid from the seminal vesicles mixes with the sperm, providing nutrients and enzymes for sperm viability. Additionally, it is the target of the vasectomy procedures, where it is cut and sealed to prevent sperm from reaching the urethra, serving as a form of male contraception.
In figure 28.2, we can see the parts of the scrotum. These are the parts that support the scrotum's structure. Internal oblique muscle, abdominal muscle aiding trunk movement, aponeuris of external oblique muscle, tendon of the external oblique muscle, fundiform ligament of penis, supports the penis, transverse section of the penis, corpora cavernosa, rectal tissue of columns, spongy urethra between spongiosum, corpus spongiosum surrounds the urethra, spermatic cord, bundle from abdomen to testicles, superficial inguinal ring, abdominal wall opening, cremaster muscle raises or lowers testicles for temperature control, ductus deferens, sperm transports tube, autonomic nerve controls involuntary functions, testicular artery, blood supplies to the testicles, lymphatic vessels carries lymph, pumpiform plexus cools blood to testicles, scrotal septum divides scrotum, epididymis sperm matures or maturation or storage, external spermatic fascia covers spermatic cord, tunica albuginea dense covering of the testicles, dartos muscle wrinkles scrotal skin, tunica vaginalis covers testicles, skin scrotum, outer layer of scrotum, internal spermatic fascia surrounds scrotum cord, raphe, seam on the scrotum. The seminiferous tubules contain two types of cells, spermatogonia and nurse cells. Spermatogonia are stem cells that develop from primordial germ cells, which enters the testicles in the fifth week of development, remain dormant during childhood, and begin producing sperm at puberty to mature. In order to advancing maturity in primary spermatocytes and secondary spermatocytes, spermatis and sperm. Sperm are released into the tubule lumen. Nerve cells, or Sertoli cells, support spermatogenesis by forming the blood-testis barrier, isolating the developing gametes from the immune system, nourishing developing cells, phagocytizing excess cytoplasms, and controlling sperm release in the lumen. In figure 28.3, this is the sagittal section of the testes in its parts: spermatic cord, blood vessels, nerves, ductus deferens, efferent ductile, body of the epididymis, origin testes, duct of the epididymis, tail of the epididymis, head of the epididymis, tunica vaginalis, parietal layer, visceral layer, seminiferous tubule, straight tubule, tunica albuginea, lobule, and septum.
Spermatogenesis is the process by which sperm cells are produced in the seminiferous tubules of the testes. Throughout spermatogenesis, the process is supported by Sertoli cells, which provide structural and nutritional support to the developing sperm cells and create a blood-testis barrier to protect them from the immune system. Spermatogenic cells are the various cells type involved in the process of spermatogenesis, which the development of sperm cells. So these cells undergo a series of stages within the seminiferous tubules of the testes, transforming it from primitive germ cells into mature spermatozoa. So first is we have the spermatogonium, and next is the primary spermatocytes, which these cells are formed from the final mitotic division of type B spermatogonia, and primary spermatocytes are diploid cell that enters the first meiotic division to reduce their chromosomes number by half. So this process results in the formation of two haploid secondary spermatocytes. Next is the secondary spermatocyte. These haploid cells are the product of meiosis 1 of primary spermatocytes. So the secondary spermatocytes quickly enter the second meiotic division to produce spermatids. Each secondary spermatocyte divides to form two haploid spermatids. Next is the spermatids. Spermatids are haploid cells resulting from meiosis 2 of secondary spermatocyte. So they undergo a series of morphological changes during spermatogenesis to become mature spermatozoa. So these changes include nuclear condensation, development of the acrosome, formation of the flagellum, and shedding of excess cytoplasm. Next is the spermatozoa or the sperm cells. So mature spermatozoa are the final product of spermatogenesis. They are specialized cells with a head containing the nucleus and acrosome, a midpiece packed with mitochondria to provide energy for motility, and the tail or the flagellum for propulsion. And once it formed, the spermatozoa are released into the lumen of the seminiferous tubules and transported to the epididymis for further maturation and storage. So throughout this process, Sertoli cell plays a crucial role in supporting and nourishing the developing spermatogenic cells. So they also help create the blood-testis barrier, which protect the developing sperm from the immune system and provides a controlled environment for spermatogenesis. So which cells secrete testosterone? So sperm transport and secrete the hormone inhibin and regulate the effects of testosterone and the FSH or the follicle stimulating hormone. So in the spaces between adjacent seminiferous tubules are a cluster of cells called the interstitial endocrine cells or the Leydig cells. So these cells secrete testosterone, the most prevalent androgen. So an androgen is a hormone that promotes the development of masculine characteristic. And for the clinical connections, the cryptorchidism is the condition in which the testes do not descend in the scrotum, and it only occurs 3% of full-term infants and about 30% of premature infants.
In humans, spermatogenesis takes 65 to 75 days. It begins with spermatogonia, which contains the diploid number of chromosomes. Spermatogonia are stem cells when they undergo mitosis. Some spermatogonia remain near the basement membrane of the seminiferous tubule in an undifferentiated state to serve as reservoir of cells for future cell divisions and subsequent sperm production. The stages of spermatogenesis. The first one is primary spermatocytes. Primary spermatocytes, like spermatogonia, are diploid, that is, they have 46 chromosomes. Shortly after it forms, each primary spermatocyte replicates its DNA and then meiosis begins. In meiosis, homologous pairs of chromosomes line up at metaphase plate and crossing over occurs. Then, the meiotic spindle pulls one chromosome of each pair to an opposite pole of the diving cell. secondary spermatocytes. Each secondary spermatocyte has 23 chromosomes. The haploid number each chromosome within a secondary spermatocyte, however, is made up of two chromatids, two copies of the DNA still attached by a centromere. No replication of DNA occurs in the secondary spermatocytes. Spermatids. In meiosis II, the chromosomes line up in single file along the metaphase plate, and the two chromatids of each chromosome separate. The four haploid cells resulting from meiosis II are called spermatids. Spermiation is the final stage of spermatogenesis where mature spermatozoa are released from the Sertoli cells. In the lumen of the seminiferous tubules, Sertoli cells play a crucial role throughout spermatogenesis, providing nutritional and structural support to the developing sperm cells. Once spermiation occurs, the mature spermatozoa are transported through the male reproductive tract, starting from the seminiferous tubules to the epididymis, where they undergo further maturation and gain motility.
Now let's talk about the spermatogenesis. Spermatogenesis is the process by which sperm cells are produced in the testes. It involves several key events and stages that leads the formation of mature cells. Now that we have set the stage, let us begin by understanding the fundamental difference between diploid and haploid cells. Diploid cells, known as spermatogonia, possess 46 chromosomes, symbolized as 2N. In contrast, haploid cells, called spermatids, contain only 23 chromosomes, symbolized as N. This reproduction in chromosomes and number is crucial for the successful fusion of sperm and oocytes during fertilization. Moving forward, we encountered a pivotal stage in spermatogenesis known as a spermiogenesis. This stage involves the maturation of spermatids into fully functional sperm. During spermiogenesis, spermatids undergo significant transformations, including the development of acrosome, a condensed nucleus, flagellum for motility, and mitochondria to provide energy. Additionally, excess cytoplasm is eliminated through the assistance of nerve cells. Now, let's explore the outcome of meiosis in spermatogenesis. As the cells progress through meiosis, some spermatogonia remain near the basement membrane of the seminiferous tubule, serving as a savior for future cell divisions and subsequent sperm production. The remaining spermatogonia differentiate into primary spermatocytes, which are diploid cells with 46 chromosomes. Meiosis I then occurs, resulting in the separation of homologous chromosomes and the formation of secondary spermatocytes, each containing 23 chromosomes with two chromatids. Meiosis II follows, with the chromatids separating and ultimately producing four haploid spermatids, each with 23 chromosomes. Throughout the development of the sperm, cytoplasmic bridges play a crucial role. These bridges connect the developing sperm cell, allowing for synchronized sperm production. This synchronization may provide survival advantage for sperm carrying either the X or Y chromosome, ensuring the production of a balanced number of each type. Mature sperm exhibit a highly specialized structure. They measure approximately 60 micrometer in length and possess a flattened, pointed head and a tail. The head contains nucleus with 23 chromosomes and the acrosome filled with enzymes necessary for fertilization. The tail consists a neck with centrioles, a middle piece with spiral mitochondria for energy production, a principal piece, and an end piece. Sperm production is a continuous process with approximately 300 million sperm completing spermatogenesis each day. However, the lifespan of ejaculated sperm with the female genetical tract is relatively short, typically surviving on less than 48 hours.
Testosterone and the high testosterone both bind to the same androgen receptors, which are found within the nuclei of target cells. The hormone receptor complex regulates gene expression, turning some genes on and others off. Because of these changes, the androgens produce several effects like prenatal development, wherein before birth, testosterone stimulates the male pattern of development of genital system ducts and descent of testes. The high testosterone stimulates development of general genitalis. Testosterone also converts it in the brain to the estrogens, which may play a role in development of certain regions of the brain in males. Another one is the development of male sexual characteristics. At puberty, testosterone and the high testosterone bring about development and enlargement of male sex organs and the development of masculine secondary sexual characteristics. Secondary sex characteristics are traits that distinguish males and females but do not have a direct role in reproduction. This includes muscular and skeletal growth that result in wide shoulders and narrow hips, facial and chest hair and more hair on other parts of the body, thickening of the skin, increased sebaceous gland secretion, and enlargement of the larynx and consequent deepening of the voice. The third one is the development of sexual function. Androgens contribute to male sexual behavior and spermatogenesis and to sex drive in both males and females. Keep in mind that the suprarenal cortex is the main cause of androgens in females. Lastly, stimulation of anabolism. Androgens are anabolic hormones, which means they stimulate protein synthesis. This effect is obvious in the heavier muscle and bone mass of most men as compared to women. A negative feedback system regulates testosterone production. When testosterone concentration in the blood increases to a certain level, it inhibits the release of GnRH by cells in the hypothalamus. As a result, there is less GnRH in the portal blood that flows from the hypothalamus to anterior pituitary. Gonadotrophic cells in the anterior pituitary then release less LH, so the concentration of LH in systematic blood falls with less stimulation by LH. The interstitial and the endocrine cells in the testes secrete less testosterone, and there is a return to homeostasis. If the testosterone concentration in the blood falls too low, however, GnRH is again released by the hypothalamus and stimulates secretion of LH by anterior pituitary.
The male reproductive system contains a series of ducts that transport sperm from the testes to the urethra for the ejaculation. Let's take a closer look at this pathway. The seminiferous tubules within the testes are where sperm are produced. Through the process of spermatogenesis, the sperm then travel from the seminiferous tubules to the straight tubules into the retetestes, which is a network of ducts. From the retetestes, the sperm enter the efferent ducts, which carry them to the epididymis. The epididymis is a tightly coiled tube about 6 meters long, where the sperm mature and are stored. The epididymis connects to the ductus, or vas deferens, also known as the vas deferens. The ductus deferens ascends through the spermatic cord and into the pelvic cavity. where it loops over the ureter and descends along the posterior surface of the urinary bladder. At the base of the bladder, the ductus deferens joins with the duct from the seminal vesicle to form the ejaculatory duct. The ejaculatory duct then passes through the prostate gland and joins the urethra, which is the common passage for the both urine and semen.
There are three functions of accessory male genital gland secretions. First is the seminal gland secrete seminal fluid, an alkaline viscous fluid that helps neutralize acid in the female genital tract, provides fructose for ATP production by sperm, contributes to sperm motility and viability, and helps semen coagulate after ejaculation. Second is the prostate secretes prostatic fluid, a milky slightly acid fluid that contains enzymes that break down clotting proteins from the seminal glands. And third, the bulbourethral gland that secrete an alkaline fluid that neutralizes the acidic environment of the urethra and mucus that lubricates the lining of the urethra and the tip of the penis during sexual intercourse. In figure 28.9, we can see the location of several accessory male genital glands such as the prostate, urethra, and penis. While the male urethra has three subdivisions, which are the prostatic, membranous, and spongy urethra, which will be discussed later. In the posterior view of accessory male genital glands, we can see different names that indicate what type and function they provide. Let's discuss them one by one. The left ureter is a muscular tube about 25 to 30 cm long that extends from the renal pelvis of the left kidney down to the urinary bladder. The function of this is that the ureter is to transport urine from the kidney to the bladder through peristaltic contraction of its muscular walls. This process ensures a continuous flow of urine regardless of body position or activity. Next is the hip bone or cut. The hip bone is also known as the oscussae, which is a part of pelvic girdle. It is composed of three fused bones, the ilium, ischium, and pubis. In anatomical diagrams, the hip bone is often shown cut to expose the internal pelvic structure. The function of the hip bone it provides structural support for the upper body protection for pelvic organs and serves as an attachment site for various muscle and ligaments. It also forms the acetabulum, the socket for the femoral head, which is crucial for hip joint articulation. Next is the prostate. The prostate is a walnut-sized gland located below the bladder and in front of the rectum. It surrounds the prostatic urethra, the part of the urethra that passes through the prostate. The function of this is that it produces seminal fluid, which constitutes about 30% of the volume of semen. This fluid contains enzymes, zinc, and citric acid, which helps to nourish and protect sperm, enhance sperm motility, and facilitates their transport. Next is the prostatic urethra. The prostatic urethra is the portion of the male urethra that runs through the prostate gland. It is approximately 2.5 cm long. Its function is it serves as a conduit for urine from the bladder during urination and forms semen during ejaculation. The prostate urethra also receives an ejaculatory duct where the sperm and seminal fluid milks. The next is the membranous urethra. The membranous urethra is a short, narrow segment of the urethra about 1 to 2 cm long, located between the prostate and the penis. It passes through the urogenital diaphragm, a muscular part of the pelvis. The function of membranous urethra is that this part of urethra is surrounded by the external urethral sphincter, which provides voluntary control over urination. It is the least distensible part of the urethra. Next is the crus of penis. The crura of the penis are two elongated structures that forms the posterior part of the corpora cavernosa. Each crus attaches to the ischium ramus of the pubic bone. The function of this is that crura anchor the penis to the pubic arch, providing structural support during erection. They are covered by the ischiocavernosus muscle, which helps to maintain erection by compressing the crura, restricting blood outflow. The bulk of penis is that expanded posterior part of the corpus spongiosum located to the base of the penis. It is situated between the two crura. The function of this is that it expels urine and semen from the urethra through construction of the bulbospongiosus muscle. This muscle also aids in ejaculation by increasing pressure within the spongy urethra. The corpus spongiosum penis is that cylindrical column of erectile tissue surrounding the spongy urethra. It runs along the underside of the penis and extends from the bulb to the glans. The primary function of the corpus spongiosum is to prevent the urethra from collapsing during erection, ensuring one channel for semen and urine. It also contributes to the erection by filling with blood. Next is the right ductus deferens. The ductus deferens, also known as the vas deferens, is a thick-walled tube about 45 cm long. It starts from the epididymis and it ascends into a pelvic cavity, looping over the ureter and descending to the ejaculatory duct. The function of this is that the ductus deferens transfers mature sperm from the epididymis to the ejaculatory duct during ejaculation. Its thick muscular walls facilitate sperm movement through peristaltic construction. Next is the ampulla of ductus deferens. The ampulla is the widened terminal portion of the ductus deferens located to the prostate gland. The function of this is that the ampulla serves as the storage site for sperm and adds secretion to the ejaculate, enhancing sperm viability and motility. Next is the seminal gland. The seminal glands are paired structure located posterior to the bladder and anterior to the rectum. Each gland is about 5 cm long and has a coiled tubular structure. The function of these is that these glands produce seminal fluid, which makes up about 60% of the semen volume. This volume contains fructose, proteins, enzymes, and prostaglandins, which provides energy for sperm and facilitates their movement and function. Next is the seminal gland duct. Each seminal gland duct emerges from the seminal vesicle and joins with the ductus deferens to form a ejaculatory duct. The function of this is that the duct transports seminal fluid from the seminal vesicle to the ejaculatory duct, where it mixes with sperm from the ductus deferens. Next is the ejaculatory duct. The ejaculatory ducts are formed by the union of the ductus deferens and the seminal vesicle ducts. They are about 2 cm long and pass through the prostate to empty into the prostatic urethra. The function of this is that these ducts convey sperm and seminal fluid into the prostatic urethra during ejaculation. Next is the deep muscle of perineum. These muscles are located in deep perineal space and include the deep transverse perineal muscle and the external urethral sphincter. The function is that the deep muscles of the perineum support the pelvic organs, maintain continence, and contribute to the stability of the pelvic floor. Next is the bulbourethral gland. It, it is small, pea-sized glands located beneath the prostate near a base of penis. This function is secrete a clear mucus-like pre-ejaculate fluid that lubricates the urethra and neutralizes any residual acidity, preparing the urethra for the passage of sperm. Next is the corpora cavernosa penis. The corpora cavernosa are two cylindrical columns of erectile tissue that runs along the length of the penis. They are encased in a taut fibrous sheath called tunica albuginea. The function of this is that these structures fill with blood during sexual arousal, leading to penile erection. Penile erection. The corpora cavernosa provides rigidity and support for sexual intercourse. The spongy urethra or penile urethra is the longest segment of male urethra running through the corpus spongiosum from the membranous urethra to external urethral orifice to the tip of the glans penis. The function of this is that the spongy urethra allows the passage, the urine of semen, out of the body. It serves about excretory or reproduction function. Ensuring smooth and efficient activities such as urination, semen, generation, and ejaculation, each of these structures is essential to the male urinary and reproductive system.
Welcome to the presentation on accessory male genital glands. We will explore the seminal glands, the prostate, and the bulbourethral glands, which secrete most of the liquid portion of semen. The ducts of the male genital system store and transport sperm. However, the accessory male genital glands secrete most of the liquid portion of semen. The accessory male genital glands include seminal glands, prostate, and bulbourethral glands. Each gland plays a crucial role in the production and secretion of seminal fluid. First is the seminal glands. The seminal glands, also known as seminal vesicles, are convoluted, pouch-like structures located posterior to the base of the urinary bladder and anterior to the rectum. They secrete seminal fluid, an alkaline, viscous fluid containing fructose, prostaglandins, and clotting proteins. The alkaline nature of the seminal fluid neutralizes the acidic environments of the male urethra and female genital tract, protecting sperm. Fructose provides energy for ATP production in sperm. Prostaglandins enhance sperm motility and viability and may stimulate smooth muscle contractions in the female genital tract. clotting proteins help semen coagulate after ejaculation, preventing sperm leakage from the vagina. Seminal fluid from these glands constitutes about 60% of the semen volume. Next is prostate gland. The prostate is a single donut-shaped gland about the size of a golf ball, located inferior to the urinary bladder and surrounding the prostatic urethra. It secretes a milky, slightly acidic fluid that contains citric acid, proteolytic enzymes, acid phosphatase, and seminal plasmin. Citric acid in prostatic fluid is used by sperm for ATP production. Proteolytic enzymes, including prostate-specific antigen, break down the clotting proteins from the seminal glands. The function of acid phosphatase is unknown. Seminal plasmin, an antibiotic in prostatic fluid, helps reduce bacterial presence in semen and the lower female genital tract. Prostatic secretions make up about 25% of the semen volume, enhancing sperm motility viability. Lastly is bulbourethral glands. The paired bulbourethral or upper glands are pea-sized and located inferior to the prostate on either side of the membranous urethra. During sex arousal, this secretes alkaline fluid into the urethra that neutralizes acid from urine, protecting sperm. They also secrete mucus that lubricates the end of the penis, the lining of the urethra, reducing sperm damage during ejaculation. The alkaline fluid from the bulbourethral glands protects sperm by neutralizing acidic urine residue in the urethra. The mucus secretion lubricates the urethra, the tip of the penis, reducing sperm damage during ejaculation. Some males release a drop or two of mucus in sexual arousal and erection. In conclusion, the accessory male genital glands are essential for producing and secreting seminal fluid, which nourishes and protects sperm, enhances their motility and viability, and ensures their safe passage during ejaculation. Thank you for your attention.
Hello, my name is Rochelle, and the topic that we're assigned to our group is the male reproductive system. My part is the semen and penis. That's the topic that I'm gonna report, and I will start with the semen. Here, as you can see on my screen, I have the semen and its composition. We have spermatozoa and seminal plasma. Spermatozoa is the male reproductive cells produced in the testes and responsible for fertilizing the female egg, while the seminal plasma is the fluid portion composed of secretions from the seminal vesicles, prostate gland, and vulvarethral glands. And this it includes these three. Fructose, it provides energy for sperm motility, and the prostaglandins, it stimulates uterine contractions to aid sperm movement. And the last one, alkaline pH, it neutralizes the acidic environment of the female vagina, protecting the sperm. Now let's gonna talk about the function of the semen. Uh, we have here two as well. First, reproductive role. Um, Semen carries sperm into the female reproductive system to, of course, enable fertilization. And the second one is the nourishment and protection. It actually provides nutrients and a suitable medium for sperm viability and mobility. Now we will talk about the health issues that may occur to semen. We have zoospermia and semen quality. First, the azoospermia. It is the complete absence of sperm in the ejaculate, and it actually leads to infertility. The next one is the semen quality. Um, it has factors like volume, motility, and morphology of sperm that are critical for fertility. And those are lifestyle factors such as environmental exposures and also medical conditions can affect semen quality. Now we're done with the semen part. So now we're going to talk about the functions of the penis. We have here three. First, reproductive role. It means that penis, it is actually the one that delivers the semen to the female reproductive tract during the intercourse. The second is the urinary role. It excretes urine from the bladder through the urethra. And the last one, erectile function. It achieves erection through blood engagement, enabling sexual intercourse. And here, as you can see on this slide, semen has another two functions. It's because the semen is an external organ of the male reproductive system, and I think it's really has a lot of functions. These are the selected ones, the ones that I selected. The first one is the sexual intercourse. So we all know this. During erotic stimulation, the penis undergoes erection, becoming engorged with blood. Following emission, ejaculation can occur, whereby semen moves out of the urethra through the external urethral orifice. And then finally, the penis undergoes remission, returning to a flaccid state. And the second one, micturition. This is where the penis also has an important urinary role. It contains the urethra, which carries urine from the bladder to the external urethral office, where it is expelled from the body. Now, what are the parts of the penis? Here we have the anatomically picture of the penis and anatomically divided into three parts. First, we have the root, the yellow part. It is the most proximal fixed part of the penis, and it is located in the superficial perineal pouch of the pelvic floor and is not visible externally. The root, it contains three erectile tissues and two muscles. The next one is the body, which is the color green. It is the free part of the penis located between the root and the glands. It is suspended from pubic symphysis. It is composed of three cylinders of erectile tissues, two corpora cavernosa, and the corpus spongiosum. And the last one, which is the glans, the color blue, it is the most distal part of the penis. Um, It is a conical in shape and is formed by the distal expansion of the corpus spongiosum. This contains the opening of the urethra, termed the external urethral office. Now, here on the next slide, you will be able to see the anatomy of the erectile tissues we have in the penis. The green one is the corpora cavernosa, which what I have discussed. And then the second one is the yellow corpus spongiosum. Okay, on this part, we're going to talk about the muscles of the penis. There are actually four muscles located in the root of the penis. So these four muscles can only be seen
specifically in the root of the penis, which is the most proximal part anatomically and also not so visible. And yeah. Okay, so we have here tissues. First is bulbous spongiosus. This is actually times two. We only have two here in the PowerPoint, but times two, so it's going to be four. These are the four muscles of the root of the penis. Let's go back to the bulbous spongiosus. It is associated with the bulb of the penis. It contracts to empty the spongy urethra of the residual semen and urine. The anterior fibers also aid in maintaining erection by increasing the pressure in the bulb of the penis. And the second one, this is also times two, that makes the muscle of the penis four. We have here is chiocavernosus. It is it surrounds the left and right crura of the penis. It contracts to force blood from the cavernous spaces and the crura into the corpora cavernosa, and this also helps maintain erection. And the last slide, it's all we have here, the ligaments. We have two as well. And the root of the penis is supported actually with these two ligaments, and they are attached to the surrounding structures. First is the suspensory ligament. It is a condensation of deep fascia, and it connects the erectile bodies of the penis to the pubic symphysis. And the second one is the fundiform ligament. It is a condensation of abdominal subcutaneous tissue. It runs down from the linea alba surrounding the penis like a sling and attaching to the pubic symphysis.